Commander Skanchy, and David Amspacher. So with that, we're going to go ahead and get started with our first question for Dr. Wu. What is a simulated partially dissolved drug substance sample? And Dr. Wu, okay, we might move on to David Amspacher for your first question. And David, your question is going to be, as process validation is often not completed at the time of DMF review, is it acceptable to provide a commitment to complete process validation and submit process validation summary in response to deficiencies raised during completeness assessment, incomplete comments letter, or CMC quality review? Complete response letter.
Hi, and thank you for your question. First off, I'd like to make sure that we know that before any drug product is shipped or distributed, the process validation has to be done. That being said, as far as the DMF goes, you can give a commitment to complete the process validation before the DMF is adequate. But that makes it a lot more difficult because we use the information in your process validation to do our review on the DMF. So we're going to ask you for that information.
Great, thank you. And our next question will be for you as well. The agency has a long-standing practice of accepting 3 batches as validation batches for API and FDF. Your presentation seems to contradict that. Any thoughts if this is the way the agency is thinking now?
Well, the most important thing about how many validation batches to do is the phrase statistical metrics. So basically, you have to have enough runs to have a statistical significance. Many times, 3 is enough because a lot of times 3 is the minimum number you have to get a standard deviation and a variance. It may take more to get what we need as far as to make sure that everything runs good. Most often it's 3, but it could be more. That's kind of the point of that.
Okay, great, David. And we have another question for you. Is any information regarding the use of recovered solvents and materials expected to be provided in section 3.2.S.2.5? Do we need to validate the manufacturing process again if we plan to use recycled solvents or add an additional 1 batch using recycled solvent to the original validation plan?
Thank you for your question. The biggest thing with recycled solvents is a specification for the recycled solvents. A lot of times we'll see where they say, hey, we're using, we're recycling the toluene, but they don't tell us how they're making sure it's pure. So the biggest thing is to make sure you have a specification for the recycled solvent. And if you know you're recycling solvents and it's part of the process to begin with, we'd like to see that in all of your validation batches if it's possible. If you're adding it later on, you don't have to revalidate, but we would like to see a one batch that you have run with the recycled solvents, and we want to see some stability data on that particular batch.
Great, thank you so much, David. Our next question will be for Dr. Wu. What is a simulated partially dissolved drug substance sample? Okay, and I think we will actually move on to another question for Dr. Gaddam. Dr. Gaddam, does the FDA accept the use of pyrolysis gas chromatography as a test method technique to determine the degree of cross-linking in cross-linked API polymers?
Yes, I mean, in the complex API sameness, there are several structural fragments need to be showed as the same. So this is one of the method can be used. It's not the only method to be used. So it's one of the method it can be used. Yes.
Great, thank you. And our next question will be for you as well, Dr. Gaddam. After approval of an ANDA, if an ANDA applicant wants to submit an alternate API, does all of the characterization done at the time of original submission for API need to be repeated for an alternate API? Or is referencing the DMF of the alternate API supplier for these drug substance characterizations, is that enough?
Thank you for the question. I think there is some clarification needed at ANDA, but at the DMF perspective, all the characterization and the API sameness should be shown, and the additional ANDA information. I think additional characterization done at ANDA level also, depending on the requirement. Thank you.
All right, great. Thank you for that answer. And we have another question for you, Dr. Gaddam. Slide 11 asks for 3 batches of the reference listed drug to be evaluated. This morning, only 1 batch of API was recommended for comparison. Which one is correct?
Thank you for this question. So in complex APIs, so basically we need statistically significant data is to be shown. So to show minimum, I mean, 3 batches we recommend, but it should be multiple batches. At least 3 batches are needed to show sameness. If we show 1 batch, we don't know how the manufacturing process is done. So based on statistical values, at least 3 batches are recommended for showing API sameness for complex APIs. If more is good.
Great, thank you so much. Our next question is for David Skanchy. If a USP monograph contains a limit for unspecified impurities, which is higher than the ICH Q3A identification threshold, which limit ultimately applies to unspecified impurities?
Yeah, so it's the ICH Q3A identification threshold limit that you want to use, not the monograph limit for unspecified impurities, the ICH limit.
Great, thank you. And our next question is for you as well. If there is a USP monograph for the API, does the impurity specification need only to meet the USP monograph, or must it be reflective of the process capability? For example, if I am capable across a statistically significant number of batches to reach 0.3% or less, but the monograph only allows 0.5% or less, do I need to set 0.3% or can I align with a higher monograph?
Right, for setting specifications for the API, the monograph limit is acceptable. It would really put you in a situation where somebody with a very good process control was actually being penalized by being forced to use a lower limit in their specification. So the monograph limit is acceptable. The only time we would ever question the use of a monograph limit is if information came to light that that limit presented a safety issue. So if that's not the case, the monograph limit is fine.
Okay, great, thank you. Our next question is for David Amspacher. Does the validation process apply to excipients or just APIs?
Thank you very much for that. The validation, if you're going to be using a anything that goes into a drug, is going to be validated. So that's the short answer. Thank you.
Okay, great. Thank you, David. We have another question for you. At which stage of the development lifecycle would you recommend or expect to see process validation? After phase 2 trials or after phase 3?
That's going to be really variable and kind of be company specific, because I know a lot of companies nowadays have changed from, they used to do everything up front, and they've changed to a model where they make just enough to get through the trials to see if they're going to do it. So that's going to depend on your particular company and how big of how much you're making. So if you're only going to make a small amount in a pilot lab just to get through the phase 3 trials, and then you're not going to scale up production until after the phase 3 trials. That's when you're going to want to do your validation. Thank you.
Great, thank you so much. And as a reminder to all of our participants, you can put your questions in the Q&A pod, and we will get to as many as we can as time allows. Our next question is for Dr. Gaddam. For complex APIs while establishing sameness, how do we apply same is same from an FDA perspective? There is no guidance.
Thank you for the question. So as I know, there is no guidance to show how much same is same, but it is a case by case. So while doing the product development, so you need to identify critical quality attributes. So you need to establish the ranges, how much, what is the range it can be accepted based on the critical quality attribute safety. So is a case by case for the compound, so it cannot be same for each and every API. So it should be established based on the critical quality attributes. Thank you.
Great, thank you. And we have another question for you, Dr. Gaddam. What kind of information can we expect from a pre and a meeting? For Dr. Gaddam, does that, I'll just ask the question again. What kind of information can we obtain from a pre and a meeting? Or David Skanchy, can you take that question?
Yeah, I can answer that. So the two types of meetings that are available are the pre-development meeting and the pre-submission meeting. The meeting that is most helpful and useful for a complex API is the pre-development meeting. And the pre-development meeting is really driven by the package that the firm submits, because in that package they will include all the questions that they want answered that have to do with the API, with the drug product. So the applicant is really the driver of the content of that meeting. And again, it is the pre-development meeting where most of the complex APIs are brought up. The pre-submission meeting, which is later after the pre-development meeting, the API questions don't tend to come up too often because they've already been addressed. So, but I've participated in many pre-development meetings, and the firms that participated, I believe, found them to be very useful.
Great, thank you, Commander Skanchy. Our next question will be for David Amspacher. Manufacturer A is a DMF holder, the DMF holder. Manufacturer A transferred a critical intermediate to another manufacturer B and manufacturer B slightly modified the critical intermediate process. Manufacturer B validated transferred intermediate process. Does manufacturer A have to revalidate their remaining process using intermediate from manufacturer B?
It's a very interesting question. I think the key thing we need to think about here is manufacturer A has changed their process. So if you change your process, you have to revalidate.
Great, thank you. And we have another question for you, David. How does one conduct concurrent validation of the process step if limited quantity of material is available, for example, a rework?
It's possible to do validation on a smaller scale, but you have to make sure that it's transferable to the larger scale. So if you do a validation on a smaller scale and your commercial scale is a lot larger, you could end up having problems that you don't realize. So we prefer to see the validation done on as large a scale as possible. So that would be the short answer.
Great, thank you. And we are going to circle back to Dr. Wu. And our question for Dr. Wu is, what is a simulated partially dissolved drug substance sample? Okay, and I think we're actually going to go to Dr. Gaddam. Should the comparability studies be conducted in a GMP laboratory?
Thank you for that question. I think the study
A simulated sample can be made by the stock solution of impurities.
Something is overlapping.
Oh, I think Dr. Wu just came on, so.
Sorry, I mean,
Go ahead, Dr. Gaddam, and then we'll just
it doesn't need to be in the GMP laboratory, but the product should be obtained from GMP conditions. It's short. Okay, thanks. I can.
Thank you, Dr. Gaddam. And we'll ask one more question to Dr. Gaddam. How is the particle size compared with the reference listed drug?
For complex insoluble APIs, particle size is considered critical quality attributes. So the characterization should be done with validated analytical method. And this should be in 3-tier, and polydisperse index is also important, and that should be considered to compare. Thank you.
Thank you, Dr. Gaddam. Our next question is for David Amspacher. Do we have to submit a process validation summary for our DMF to be adequate?
Thank you very much for the question. For any drug product to be distributed, a validation has to be performed. A DMF can be adequate without the process validation being done, but it's very difficult because we use that information that is in the process validation to help justify the limits for all of your intermediates and all of your impurities. So if you submit one without it, we're going to ask for it, and it's going to make it harder for you to get your DMF made adequate if you don't have the process validation. So it can be done, but I wouldn't advise it.
Great, thank you. Our next question is for you as well. Can we use 3 sigma to set the limits in our specifications?
Sorry, could you repeat that again?
Sure. Can we use 3 sigma to set the limits in our specifications?
Yes, thank you very much. So 3 sigma is an adequate statistical method to use to set limits. So and that's actually one of the methods that we prefer. If you have, for example, say 10 runs that you've done, and you can then use all of that to do a 3 sigma analysis. That's a very good way to do it.
Great, thank you. And another question for you. Can we have a higher limit on an impurity upstream of the API if we have a spike and purge study?
Yes, you can. So spike and purge studies are one of the ways to determine if your process can purge a particular impurity. So if you have, say a limit of you want a higher limit to have more flexibility in your process, but you normally only see a lower limit. If you do a spike and purge at the higher limit or in excess of the higher limit, that can be used in your DMF.
Okay, our next question is for Commander Skanchy. Is there any possibility that the DMF would be rejected by the agency during the CA or TCIR, just like the RTR to an ANDA application? Thank you so much.
Yeah, so I'm going to assume that the asker of that question is referring to the validation data. And for the completeness assessment, the way we treat the presence of validation data is by asking the question, is the process required to have the validation done prior to approval? And so if it's a sterile process or if it's a biotechnology process, those are the cases where validation has to be in the DMF up front prior to approval. For all other processes, which is the vast majority of DMFs, you would not be required to do validation prior to approval and commercial marketing. So if the DMF came in, it was not a sterile or biotechnology process, and you did not have the validation information, you would still pass the completeness assessment. Now, here's sort of the catch with completeness assessments. If you come in with a DMF, even though you're not required to do validation, but you presented to us the validated process and did not include your validation summary, we will ask for that on completeness assessment. Not that you were required to do the validation, but since you did it, we would ask for that at the completeness assessment. And the reason is Dave Amspacher said it very well. The process validation data really helps us doing the DMF review, and we have a lot of confidence when we say that process is adequate if the validation's been done. And fortunately for us, the overwhelming majority of drug master files come in with the validated process, even though most aren't required to.
Great, thank you, Commander Skanchy. And we have another question for you. Under what circumstances do third-party contract laboratories have to obtain an EIF number with the FDA?
Well, that's the facility-related question, so I'd defer that one to our facility experts, and we'll present the answer to that one at the follow-on webinar.
Okay, great. And with that, we're going to go back to Dr. Wu, and it sounds like from our technical team, there may be a short delay. So just for our audience, just keep tuning in. I think he's going to answer the question, but it just takes a couple seconds for it to catch up. So Dr. Wu, what is a simulated partially dissolved drug substance sample? Okay, and I think we're still having an audio.
Simulated
Oh, here we go.
sample can be made by spiking the impurity to the API and dissolve the whole sample with organic solvent. After evaporation of the organic solvent and dry the sample, that can be considered as a simulated sample.
Great, thank you, Dr. Wu. Our next question for you is, when impurities mentioned in a USP monograph are not available with USP Commission in that case, is it okay to validate or verify the USP method without these impurity standards? And again, we may have a short delay.
Thanks for the questions. And if the impurities are listed in the USP monograph, we do encourage the company to find the third CRO companies, synthesize, or they can make the impurities by their own. That's our recommendation.
Great, thank you, Dr. Wu. Our next question for you is, what description of analytical procedure is expected for a compendial method?
And to be sure, we expecting is same as in-house method.
Great, thank you so much, Dr. Wu. Our next question for you is, what do you mean by system sensitivity? LOQ?
It can be either the validate LOQ or the reporting threshold, but we are expecting is the sensitivity should meet at least the reporting threshold.
All right, thank you so much, Drs. Wu and Gaddam, David Amspacher, and Commander Skanchy, for all of your time presenting and answering all of our questions. And with that, now we